Your body is composed of roughly 37 trillion cells, each shielded by a dense, oily barrier. Yet a single molecule touching the outside of that wall can trigger a massive physical reaction inside the cell. This creates a physical bottleneck. Vital signaling hormones, such as angiotensin II or vasopressin, are water-soluble. Since the cell membrane is made of lipids that repel water, these messengers ricochet away from the exterior. They carry urgent instructions, but lack the chemical makeup to enter the cell and deliver them. To bridge this gap, the cell utilizes a G-protein coupled receptor, or GPCR. This protein spans the entire width of the membrane, serving as a specialized relay station. When a hormone docks into the receptor's exterior, the GPCR immediately shifts its geometric shape on the inside of the cell. The message passes through the membrane, even though the molecule itself stays outside. By utilizing this relay, the cell transforms a static physical wall into an active communication interface. Positioned directly beneath that receptor is the GQ protein, a three-part molecular cluster. As the receptor changes shape, this cluster activates. Activation requires an energy trade. The GQ protein's alpha subunit releases a low-energy GDP molecule and captures a high-energy GTP molecule from the surrounding fluid. This influx of energy causes a physical break. The alpha subunit snaps away from the beta and gamma subunits. Now independent, the alpha subunit glides along the inner surface of the cell membrane to locate its next target. The external signal has successfully crossed the membrane and transitioned into an active, mobile messenger within the cell. The alpha subunit's target is phospholipase C, or PLC. As this diagram shows, PLC is a massive, complex enzyme anchored to the membrane. The alpha subunit binds to PLC and switches it on. Once active, PLC identifies a specialized phospholipid located in the inner membrane layer called IP2. In the central event, PLC acts as molecular scissors. It cleaves PIP2 precisely in half. The first fragment, IP3, is water-soluble, detaching from the membrane and drifting into the cytoplasm. The second fragment, DAG, is a lipid, remaining anchored and sliding laterally. A single signal has now bifurcated into two distinct tools, each engineered to operate in a different part of the cell. IP3 travels toward the endoplasmic reticulum, a massive membrane structure that serves as the cell's internal calcium reservoir. IP3 binds to specific receptor channels on this reservoir. This docking forces them open, flooding the cell with a sudden rush of glowing calcium ions. While calcium levels rise, the DAG molecule remains at the membrane. It is unable to function alone and must wait for the cellular environment to change. The newly released calcium ions surge toward the cell membrane, meeting the waiting DAG molecule. Both calcium and DAG dock into a larger complex called Protein Kinase C, or PKC. This simultaneous binding activates the kinase, driving the cell's final response. By splitting and then recombining the signal, the cell ensures that powerful effects, like the contraction of a muscle, only occur when specific biochemical conditions are met simultaneously. The logic of this sequence mirrors an emergency alert system. The hormone is the pressure of a hand pressing an alarm button, and the GPCR is the control panel translating that pressure into an internal signal. In this system, PLC is the dispatcher dividing the orders. IP3 is the messenger sent to release the water tanks, the calcium, while DAG remains at the station to coordinate the final response team, PKC. This entire coordinated sequence, from the initial touch to the final internal reaction, takes place in a matter of milliseconds. To review for your exam, Step 1. The hormone binds the GPCR,